So after this tour, we are now back into the presentation session, and we continue with a talk given by Marc Heuermann of IPK. Marc is postdoc in my group and has done during his PhD work a lot of experiments to benchmark the cultivation in the PhenoSphere, and he will report about that now in his talk. So Marc, the floor is yours.
Thank you, Thomas, and hi to the audience online. So my talk is as a title, Climate Simulation and Phenotyping in the PhenoSphere Container in PhenoCrane System. And as Thomas has teased, I am going to talk about the benchmark experiment, which was benchmarking the capabilities of the PhenoSphere to actually simulate field-like conditions. So first of all, what is the need for PhenoSphere? Obviously, the natural environment would be the field. But for example, for research purposes, there is a need of exposure of plants, especially crop plants, to relevant field-like conditions in a reproducible manner, to answer questions like for systems biology analysis, towards unraveling molecular mechanisms underlying the expression of important traits, or to test hypotheses deduced from these analyses on modulation of performance-related traits in different environmental conditions. Or what I am personally very interested in is to, in future, investigate the performance of certain genotypes under expected weather conditions of future climate scenarios in comparison to contemporary situations which we have in our natural world. So the PhenoSphere provides opportunities of overcoming limitations of typically climatized glasshouses and climate chambers. And this is when I talk about the PhenoSphere in this talk, I specifically talk about compartment 1 and 2, which you have seen in the presentation now. So these are the two compartments where we have the full climatization setup, and the PhenoSphere integrates into the IPK phenotyping infrastructure to assess plant performance in specifically designed, relevant, and field-like and fully controlled environments. And the parameters which we can control are first, obviously, the illumination. So it's a combination, which you can see here, out of metal halide lamps, ultraviolet A lamps, and differentially colored LEDs. We can mix together a color spectrum which reflects a certain daytime. For example, at midday, have more blue content, or at sunset and sunrise, a higher red content. Furthermore, we can control air and soil temperature. And especially with the air temperature, we can, if the light is not on at night, we can go as low as 5 to 10 degrees. And when the light is turned on, the lower limit is around 10 degrees and up to 40 degrees Celsius. And the system was specifically designed that we can regulate that in a very fast manner. On the other side, we also control the relative air humidity to align that with the temperature and with the light intensity. And typically on sunny days, our relative air humidity inside the system is around 30 to 40%. And during night, we can reach 90% of relative humidity. Other parameters are the wind simulation. So you can see these big black fans, which are blowing from opposing directions. We can reach up to 4 on a scale from Buffo, so simulate a moderate breeze, which has proven to be enough to induce wind stimulus on plants and to change their habitus. Furthermore, we have a carbon dioxide distribution system, which in the future is targeted to be able to provide up to 1,200 parts per million of carbon dioxide content in this atmosphere, which should be enough, at least in the current predictions, to simulate the changing climate of the next 100 years. Watering is done in an automated fashion with drip irrigation system. And furthermore, we have aerial and soil sensors with which we can monitor temperature, humidity, and light intensities throughout the system. So we have 2 compartments, 2 plant stand area, each of 116 square meter size, and a maximal load out, at least in these experiments I'm showing you, was 110 containers. So with a side view panel of the PhenoCrane, we block 1 container position, which reduces it to 54 containers, so 108 container maximum with the current setup of the system. And this is, again, the overview about the compartment 1 and 2. So you have seen there was a wall in between, which separates these 2 compartments, but we can remove this wall and combine the gross area to enable larger experiments as well. And the plants are cultivated not in pots, but in this large soil volume container of 1 cubic meter of volume. and they are stacked like this. So in the lowest level, we have the base element, which are just some pellets covered by fleece to allow drainage and avoid waterlogging. Then we have coarse gravel, coarse sand, loam, and loess, and in the uppermost layer, we use a substrate mixture, or for certain experiments, we just dug out topsoil level from the IPK field site and then put that into these containers to be as close to field environments as possible. So now I'd like to show you how we designed the climate simulation. So for these experiments, I'm going to show you we run two experiments in the PhenoSphere, and the first we call it average climate, and the second we try to do a single season simulation. So let's just assume that we have a typical week somewhere in the growing season, and we have a temperature profile from the year 2016 here in blue. So some days are higher temperature, some are lower. We have a certain range in amplitude and a certain pattern which is distributed. Another year could be the same amplitude, but a different pattern, a different day distribution. And for example, in the year 2018, it could be, again, a different pattern which we find. So for the first simulation, we calculated the daily median. So if, for example, in this case, 2017 would contribute its date on the Monday. On a Tuesday, we would take the day from 2016 and so on for a certain week. And now we would take those median days with the highest temperature profile and say they are sunny days, take those with a lowest temperature profile and use them as cloudy days. And we can also then calculate the average over all these 7 median days, which we then refer to as normal days. And in the first experiment, we then distributed them over the course of a week with 3 normal days, 2 sunny and 2 cloudy days. And in each individual week, we replicated the same pattern, but obviously the day length was adapted and the temperature profile was adapted individually for each week. And the second experiment is a single season simulation where we just take the template of 1 year, and instead of calculating medians or something, we just take the days as they come and refer to as a warmest day as a sunny day, the coldest day at the cloudy day, and then calculate again the average over these 7 now real days and deem them to be normal days. And for these experiments, we didn't rely on the same pattern or the same frequency, but we allowed that to differ. So for example, in this instance, we would have 3 sunny days, and that would be represented then in the pattern of the simulation. And if you just look at the amplitude by using real days and not median days or some average, we would expect a larger variation in the field-like simulation. So for the average climate, just to reiterate, we have the same patterning every week. And for the individual growing season simulation, we have an individual pattern and frequency for each individual simulation. So here you can see now the 7 temperature profiles of the environments where we phenotyped in. So I was growing 11 maize inbred lines from America, from Europe, and from Asia, and then phenotyping them for 4 consecutive years at the IPK field site in double row plot design. And here you can see that the temperature profiles of 2016, 17, 18, and 19 from May to mid-September in an hourly resolution. As a reference, we grew the same population in a standard climatized glasshouse in pots. And as you can see, this is the temperature profile, which is much more stable and has a lower amplitude than the environments which we find in the outside world. And then these 2 PhenoSphere experiments, one with the averaged climate and one with the individual simulation of the growing season 2016. When we just calculate the standard deviation of these hourly resolved temperature profiles over the growing season, you can see that the typical standard deviation for a field environment ranges between 5.6 and 6.9 relative to the year, and that our simulation of the year 2016 comes pretty close of the actual template year 2016. The average climate is lower than its templates, and the greenhouse has the lowest standard deviation. As you can see, we basically have no pattern and a very low amplitude change over the course of the experiments. If we now correlate these temperature profiles and see how close we actually come to the true picture, we can see that we have the highest correlation between the field 2016 and its simulation inside the PhenoSphere with a correlation coefficient of nearly 0.9. And we have a lower correlation between the year 2016 simulation and all the other field years than in comparison to the average PhenoSphere climate. So for the average PhenoSphere climate, we used, as the term refers, the average climate over 3 years, 16, 17, and 18. And so we have a higher correlation with any of the field environments than with the PhenoSphere, except for the highest correlation to the field 2016. And interestingly, this pattern also follows, so this pattern of the correlation also follows between the 2016 simulation and the field. So we have the lowest correlation between the 16 profile and the 17 profile, and the highest between 16 and 19. And the same we find with the simulation. So here we also have the lowest correlation with the field 2017 and the highest with the field 2019. And the overall lowest correlation we find between the glasshouse and any of the dynamic environments. So if we now zoom in, you can also already nicely see that by respecting the pattern of these days and their different frequencies, we are coming much closer to the template than the average climate. And we will now have a look at the R-square of the correlations. We can pretty much interpret that around 80% of the found variation in any of these, between these environments, we can predict by the other one. And this is much lower if we now include the average climate in the PhenoSphere, where we only can predict 50% of the found variation with either the year 2016 or the simulation. So by averaging, we lose dynamics, and we conclude from that we need to model single days and especially respect their patterns as they occur in which orientation in the time of the year. And this becomes also pretty clear, the effect of that, if you look at the phenotypes. So you can see here the greenhouse, the average climate, the simulation of the year 2016, and here the field phenotype of the year 2016 of 3 genotypes. And these photos were taken around 47 to 50 days after sowing, so it's quite hard to synchronize 7 years of experiments. And what you can see by naked eye, that these plants are basically all at the same days after sowing. And in the greenhouse, plants are much, much larger, have a slender statue. They didn't invest into their stem, so we have to fixate them, otherwise they would fall over. With having the input of wind movement, the plants become much more sturdy already in the averaged climate, but they are larger. And running the simulation of the single season, we are pretty close now to field phenotypes. And this is just to visually score at one time point, but I was interested into the performance over time of the environment. So I calculated the best linear unbiased estimators for each of these 11 genotypes at each time point of measurement inside each environment, and then fit a nonlinear logistic growth model onto these blues. So each of these colored curves here represents basically the growth, the overall best fitted growth performance of this population in each individual environment. And what you can see directly here is that plants grow fastest in the greenhouse. So the images were taken around here. So you can see that the plants in the greenhouse reach around 1.50 meters, 1.60 meters. In the average climate, they were already above 1 meter. And in the field environment and in the plant cultivation, in the PhenoSpheres running the simulation, it was pretty indistinguishable in plant size. And this was true for the whole growth period. So the simulation came pretty close to this blue line, which refers to the growth performance of the year 2016. And this also is true when we calculate the first derivative, so the growth speed. So when do these populations reach the maximum growth speed? And here, all the field environments and the simulation of the single year maximum growth rate is reached at around 70 days after sowing, and there's no significant differences between these time points, but there is with the glasshouse and with the average climate, which are reaching this phase much earlier in the developmental phase. So we find no difference between the inflection point for field 2016 and the PhenoSphere 2016 simulation. And we can also look at that from other traits. So here is the vegetative stage, just counting the number of mature leaves on a maize plant. And you can see very nicely that the simulation follows the field environments, and both the greenhouse and the average climate are quite artificial and developed nearly a month earlier. And the same can be found with the tasseling phase. So just here plotted are the plants of a total percentage of the population which show a tassel. And you can see that plants in the greenhouse tassel nearly 1 month earlier than the plants do in the field or in the simulation of the single year. And the plant, the PhenoSphere with average climate is intermediate, but still around 20 days earlier in flowering time. So let's come to the conclusion of this experiment. A growing season at the IPK field site can be simulated in the PhenoSphere. Plant height, developmental stage, and tassel occurrence developed field-like in the PhenoSphere, simulating the year 2016. And so to produce field-like environments, the daily amplitude is found to be important, and it was found to be too low in the PhenoSphere average climate. And furthermore, the patterning, so what day comes at what time point. So the PhenoSphere simulation of the year 2016 is an approximation, but it was sufficient to reach field-like amplitudes of climate parameters. So now I would like to give you a short overview about the PhenoCrane system. So you have already seen it in motion. The PhenoCrane is a sensor to plant system, and it was integrated just last year. And it's currently, we are trying to get it from prototype into operation mode. And we can take daily images from top and side view, and with diverse cameras can target diverse species and research questions. So on the PhenoCrane, we have 4 camera systems, a high resolution RGB imaging system where we can capture architectural and color related traits in comparison to the LambdaTech system. We have a 3D laser scanner where we can compute the precise canopy height of a container. The FluoCam where we can measure kinetic chlorophyll-fluorescence, as we also do in the LambdaTech systems, and this hyperspectral camera with visible and near-infrared range, which is only present in compartment 1 currently. So we hadn't enough money now to invest it into compartment 2. And furthermore, the whole system is monitored with webcams to ensure crane movement and environmental sensors. So just short overview about the performance of the system. We have a 12 megapixel camera for the RGB camera, and we can synchronize it here with the FluoCam panel so that we get homogeneous illumination. And this is the RGB camera, and this is the FluoCam camera. And there we can extract in the future individual plants and parameters which are related to the morphology of a plant. You can see it in action, but I think we have already seen it better in real time in the system now. So basically 50 container can be phenotyped in about 40 minutes. And here's a little video from 2 months of phenotyping. So you can see we can adjust in height, and every time this PhenoCrane manages to precisely go to the correct position and take stable images. And here we can track these rapeseed plants from Dominic until maturity, basically, until silk state. That is images from last week. So furthermore of the FluoCam device, where Dominic and Henning are currently working hard to translate the protocols which we have for the FluoCam devices in the LambdaTech systems to now also utilize this knowledge for the PhenoCrane and be able to phenotype dark and light adapted plants inside the system. So the FluoCam is next to the RGB camera, and here you can see actinic light source 1 with orange light. So this is how it would look in live view if you would control that on a control panel. And we have an actinic light source in the range of red and orange space. We have cool white LEDs for actinic light source 2, and the saturating pulse can reach intensities of nearly 6,000 micro-Einstein. And we have an additional light source to complement the far red spectrum. So the 3D laser scanner is working basically like an office scanner. So it's scanning from here on a rail system and then scans iteratively the whole area. The parameters can be projected leaf area, plant height, or compactness, and we can use the data from the FluoCam, and the RGB measurements can be fitted to 3D models as surface texture to get nice images. Scanning time is roughly 2 minutes per container, but these scans have to be at night because it's very susceptible to light impurities, so it has to be in absolute darkness. Okay, so this is how it looks like. On compartment 2, Dominic has produced nice images, and you can see already a lot of details on these small rapeseed plants, and also here you can see the droplet irrigation system. So we are quite hopeful that we can utilize it to its full potential. Then we have the hyperspectral camera. I can't tell you much about that because we are still working to get it in operation mode. I think Dominic told me just recently it works now. So we have the calibration plate. The system goes there, calibrates itself, and then use that as a reference to normalize the hyperspectral images taken. And again, I would like to mention that this is only installed currently in the compartment 1. For compartment 2, we sadly don't have a hyperspec. And last but not least, we have the side view panel as well, which is looking like that. So it can be semi-automatically mounted to the crane. And then we can also capture side view images of plant sizes of up to 2.8 meters. But you have to keep in mind that the container layout obviously needs to change that, because with the current setup with densely packed containers, we would not be able to move through the system. So you need to plan that in advance and design a different kind of experiment and have a more loose container setup to enable movement. Okay, and with it, I would like to acknowledge a lot of people, especially Thomas, Astrid, Dominic, Henning, and Kerstin, but also all the people in the background, which you don't see, all the members of the research group Heterosis, ADP and APP, the gardeners, technicians, engineers, and bioinformaticians who invest a lot of time into maintaining the stuff and taking care of plants. And without them, all this stuff would not be possible. Obviously, I want to thank the funding agencies, and I would show you that we have a digital tour of the IPK on the website. You can find that there. So don't be afraid. It's the first few links are in German, but the videos are subbed in English, so you can look. It's a VR camera. And obviously, I would like to thank you for your attention.
All right, thank you very much, Marc. And there's already the first question by Hendrik.
Yeah, Marc, this is very nice. Temperature is incredibly important, but yes, light is important as well. And you haven't talked about light at all. You said you could achieve 1500 micromole at 10 degrees or so, right? But what was the daily light integral that you applied during this course of the season? And should we conclude from the fact that you probably won't get the exact light integral, that temperature is more important than light to... So light was obviously adapted. So I don't have the time now to show you all the slides, but for normal days, we used 1500 micro-Einstein as maximum and simulated 5 hours of light fluctuations over the day. Obviously, the light color was mixed together, that we have more red light in the early time. For the sunny day, obviously we have the same light intensity, because regardless of cloud cover, sun distance to Earth is the same, but we reduce the amount of fluctuation. So we basically simulated clear skies for sunny day. And for cloudy day, we simulated fully cloud cover. So a strong reduction in light intensity. Metal halide lamps were turned off, only LEDs were on, and ultraviolet with reduced intensity. So we reached around 400, 450 micro-Einstein on these days. And we furthermore simulated some light flecks. So 8 hours of light flecks and fluctuations distributed throughout the period. And because we are simulating a whole growing season, we also adapt every week the day lengths, both for temperature and also for the light simulation, to shorter days, for longer days until the 21st of June, and then reducing the day lengths again until September, October. It sounds very good, but can you tell me, outside you will get on an average season 35 to 40 moles per square meter per day, right?
Yes.
Did you achieve that in your system?
No, we don't achieve the same light intensity as we do outside. So the numbers of 1500 are on container level. So if the canopy grows, especially with maize, and you measure the intensity at canopy height, obviously the closer you come to the light source, the higher the intensity becomes. So I think something like 1700 we can achieve if you are with high maize plants. But yes, we don't achieve the same light intensity as we do in the outside world. But still, you can see that at least plant height and developmental speed were not affected by this little lack of percentage in light intensity.
So your conclusion would be that temperature is actually more important, or it's more strict to consider the right temperature than it is to completely consider the right light intensity.
From my experiments, I would say yes, that the light was basically sufficient, which we applied here. And the temperature component and the patterning of days that had a much greater effect on the actual phenotype than the light.
Soil temperature?
Pardon?
The soil temperature, did it track the...
The soil temperature we also tracked, but so we monitor the soil temperature in the outside world and found that it basically follows the season with a lower reduced amplitude, and pretty much the same pattern we find inside the system if we don't apply any cooling at all. So the soil volume is large enough that it has a retardation in heating. So you see the daily pattern and you see a seasonal pattern, but with the same reduced amplitude as we see outside. But obviously, to give a final conclusion, we still have to invest dedicated experiments where we manipulate soil temperature properly. Yeah.
Thanks.
You're welcome.
Yeah, and I would also encourage the people in the video conference to raise their hands. Oh yeah, it's coming. All right. But first here, yeah.
Yeah, I have one question in regards to the preparation of the soil that you mentioned. You got soil from the field, that's right?
Yes.
And I would say, how did you exclude the effect of microbiome for each container? I mean, if you didn't. Yeah, fungi or maybe weed.
So normally we mix all the soil together and then sample from that to ensure that basically every container gets the same from the same sample. So we don't take one inch out of the field and put it in one container. We sample it all together, mix it. And I think also the field steamed. It was steamed, the field soil. But one experiment I didn't tell you, so there was also a control experiment between the average climate and the simulation experiment, where I just used two genotypes in every container to estimate positional effects. So we had a setup where we reused the soil as it was. Then we just exchanged the top layer and we totally exchanged the whole volume. And what I found is that we have an effect, a very mild effect. So if you do total variance composition at 2 or 3 time points over the whole growing season, I found that 2 to 3% of the variation can be attributed to the row effect, so the direction of wind, of the wind movement, and we have a very mild of 1 or 2% of the total variance of pre-culture. So if we reuse the soil and we have other genotypes inside, but the soil treatment alone, at least for these kind of maize experiments, didn't show any contribution, any significant contribution to the total variance decomposition.
Yeah, but I mean, still it could be some effect of the weed.
Most definitely, but
yeah.
at least from phenotyping plant height and development stage, it's not detectable in the models. But I would give you obviously it is there, yes, but we still are not with these kind of phenotyping. Maybe with the hyperspec, we have the precision to find differences.
Yeah,
at least it was homogeneous enough, and through the steaming, we reduced again the amount of microbes.
And the other question was about the NIR camera that you mentioned. Which traits or what's the output of this camera, and which traits you can measure with this camera?
Means the hyperspectral camera?
Yes, that's right.
So currently, as I mentioned, I can't show you any images because we are just setting up the system. But typically with hyperspectral camera, if you train it properly, you should be able to score biotic, abiotic stresses, nutrient content. But obviously, this needs investment of a lot of training of these systems. But as other people are using that and are quite successful in texture, color of beans or whatever, so we are quite hopeful that we can also use it to this potential.
Thank you.
You're welcome.
So here's one question in the chat and another one by Zhu Wei Cheng. First in the chat, what was your approach to simulate the air velocity around the crop? Yeah, so the wind movement, we are quite lucky because as specifically said, we can simulate an IPK field scenario. So we also tracked the wind movement over all these years because our weather station tracks that. And what we found is that the wind comes from 2 opposing directions and changes every couple of days, basically. And there's not much in between. And with 2 opposing fans, we can simulate that. But we ramped it over the day. So we had the highest wind intensity at midday and the lowest wind intensity at night. But even at night, we had wind simulation.
So I think we went for 50% of maximum speed. That should translate to something like 3 to 4 meters per second, which was found to be, at least over the years, to be in the average wind range. So we can't simulate thunderstorms or extreme events, but we should be able to simulate what is typically found on a daily basis on a field in regard of wind speed.
Okay, then first, Suwei Chen, please unmute yourself and ask your question.
Can you hear me?
Yes, I can.
Okay, thanks you. Thanks for this the great talk, and it is amazing how capable that the PhenoSphere in IPK is. My question is actually a little bit more philosophical, because actually we know there are infinite possibility of combination of fluctuations, and could be temperature, could be light, could be also the combination of the both. In the future, if someone would like to conduct an experiment, which kind of combination of fluctuation that you would suggest?
It's a good question. So what I can say now, from the combination we choose here, with an approximation of simulating 3 days and 3 different light patterns and replicating that, we came pretty close, at least for maize, to what we have found in the environment. But then again, I think with the LED, with a combination of metal header lamps and LEDs, we should be able to simulate also the frequency changes which we found in nature. So having basically a baseline light intensity supplied by the metal header lamps, and then turn on and off the LEDs to simulate rapid cloud movement, for example. That should be no problem. If you just measure on a field and you can see there's a cloud coming, light intensity can drop by up to 1,000 micromole or more even now. And that is something which we should be able to simulate. And I think, we cannot simulate every combination, but for example, for future climate change, we could repeat these kind of experiments with projected temperature change and with carbon dioxide content, and then hopefully get some meaningful results out of that. But maybe Thomas want to comment on that?
We recorded the natural fluctuations, so by seconds, and then tried to mimic what we've seen in these profiles, and however translated actually by eye into what is possible, like sinus waves with different amplitudes and different period lengths into the system. There is still ample opportunity to improve and get further, but I think we try to mimic the amplitude of changes, and also kind of the frequency of changes that we typically see when we have sensors and record light fluctuations outside. Okay, there are 2 more questions. One in the chat. Oh no, there are even more. No, we're running out of time here. Have you collected the end dry biomass of your plants, ideally per organ, and how much variation have you seen?
So we did do that for the PhenoSphere experiments, at least for the average comparison to the greenhouse. But for these, I mean, I didn't introduce now the population. So the population was chosen to be extremely diverse in terms of how much leaves they make and what the flowering time is. And so if you let a population mature and wait until the last genotype is mature on the field, some others are nearly rotten. So I never thought that in these at least in the experiment we currently designed, it was not very meaningful to utilize this information later. For example, there is between some genotypes, there's over a month or month and a half difference in when they reach final plant height. So if you now, on what time point do you sample, and then you cannot phenotype them anymore if you cut them. So yeah, basically I cannot give you a satisfactory answer about that in this context, because the experiments, I think, are not suitable to perform this.
Okay, then Abul, please unmute yourself.
Thanks, Mark, for your nice presentation. So my question is, especially for the temperature, so when you fluctuate from day to day, how much difference are between the days? Because I am concerning about the stresses of the plants. Maybe I missed that. So how much unit you recommend to be in between the days?
So here you can see the profile. So this is not something this is basically from a measurement sensor on an hour resolution, measured from May to mid-September on the field inside the PhenoSphere, running the average climate and the single simulation. And you can see that basically the amplitude itself is a little bit reduced in comparison to the field, but it's coming pretty close. So I would not say it's only the amplitude, but it's also the patterning. For example, sometimes where there are basically days with only low temperature or with only high temperature, you introduce variation to which the plant has to invest into this adaptation mechanisms, which obviously costs energy and then reduces growth speed and comes more into the field range. So I would say that amplitude is important, but also the patterning and the time orientation, which day comes when. So I think this matters. So you have to check your target environment and then think about how to simulate that.
Thanks. Thank you very much.
You're welcome.
Okay, and perhaps final question from the chat, then we have to move on. Based on your results, what is the maize trait most strongly affected by high temperature?
I think I cannot deduce from this experiment because I didn't test specifically high temperature, but from the literature, maize optimum temperature is around 32, 33 degrees. And after that, it drops sharply in every organ development, which is, I think, according to Tardieu, quite synchronized. So I would advise you to read Francois Tardieu's paper. He has done very nice research about the temperature response of maize and other crops as well.
I'm sure Pedro knows that
Okay, sorry.
very well. All right. Thank you very much.